Dr. Steve Nadler. Steve is currently Senior VP and Head of Discovery and Translational Research at Aro Biotherapeutics. Steve's got more than 30 years of experience in drug discovery and development and translational research prior to joining Aro. As the former Executive Director and Head of Immunoscience, Immuno-Oncology and Oncology Discovery Translational Research at Bristol Myers Squibb, which is quite a mouthful, but very good title. He was solely responsible for establishing the company's Immunoscience and Immuno-Oncology Discovery Translational Research Group. And during his time at Bristol Myers, he took on roles of increasing responsibility in autoimmune and oncology therapeutic areas, where he was instrumental in the discovery and development of 5 biologic and small molecules which entered the clinic for autoimmune diseases. He also led early clinical development for 3 of these biologics. Steve was an integral member of these teams which brought both Orencia and Neurologics from discovery to the market, and he received his PhD from the University of Texas at Houston, followed by postdoc studies at the Yale Medical School. He's authored more than 80 publications and is co-inventor on over 10 patents. So Steve, it's a great pleasure to have you with us today. We really appreciate you joining us. And the title of your talk is Centyrin siRNA conjugates, a novel modality for the treatment of human disease. So thank you very much, and over to you.
Great. Thanks, Emily and Jeremy, for the opportunity to speak today. Let me just get that. What I'd like to do is share with you the work we're doing at Aro on a new protein platform called Centyrins. Specifically, we're using Centyrins to deliver siRNAs that I'll talk about. So I'll go through the background of the Centyrin platform and then talk a bit about some of the work we're doing to assess the potential for immunogenicity in vivo and in the clinic. So Centyrins are a proprietary antigen binding platform. The protein scaffold is built on the tenacin C FN3 framework. So it's a fully human framework, although about 15% of the amino acids are in the region to which we undergo immunogenesis to improve binding and alter specificity. They're small, so they're about 10 kilodaltons, extremely stable, and I'll talk more about that towards the latter part of the talk. We express the Centyrins in E. coli, so there are no disulfide bonds, there are no glycosylation sites. We have identified, however, 23 different sites within Centyrins to which we can place a cysteine and not disrupt the structure or the ability of the Centyrin to bind to its target. And at these cysteine residues that we can engineer in, we can then chemically conjugate various payloads. I'll show you in a second that Centyrins are highly specific, and it's very easy to conjugate payloads to Centyrins. And importantly, it's not a known autoantigen. So this is a diagram of the structure. So on the left is the Centyrin, again, small in size. They have both sheets and loop regions. We've established bioconjugation chemistry, so it's a very facile approach to conjugating any payload to the Centyrin at the cysteine residue using Lamide chemistry. And then we have proprietary oligonucleotides. We at Aro are specifically working on siRNAs as opposed to ASOs, antisense oligonucleotides. So we have engineered in a single cysteine, and so we do reproducibly have a single siRNA per Centyrin, and that's shown here on the left in the chromatography plot, where you see a very nice homogeneous band, and then on the right in the mass spec analysis, where you see the mass of the conjugate at the right molecular mass. So reproducibly, and we think this is very important for manufacturing, we have a DAR or drug Centyrin ratio of 1. So over the last 20 years in the field of siRNAs and antisense oligonucleotides, the majority of the drugs that have been approved target the liver. And so it's been extremely challenging to actually have extrahepatic targeting of oligonucleotides. And so that's the approach we've taken, is we're using Centyrins to deliver siRNA payloads outside of the liver to specific tissues. And we're working currently, I'll show you some data on the ability to deliver to skeletal muscle, and cardiac muscle. Now, we're also doing work on delivering siRNAs to immune cells, tumor cells, as well as some additional tissue types. Again, we're using the Centyrin to bind to a receptor. That's an internalizing receptor and can deliver the siRNA into the cell and mediate risk-dependent degradation of target mRNAs. So the Centyrin that we're working on currently is a Centyrin that binds to CD71, also known as a transferrin receptor. CD71 is ubiquitously expressed. It's important for iron uptake into cells. It's an extremely efficient receptor for taking up transferrin. Virtually every cell requires iron, and so the internalization rate is quite rapid and quite efficient. We've generated a large diversity of CD71 Centyrins to really enable us to very specifically deliver these payloads into CD71 positive cells. And we've also identified Centyrins, I'll show you in a second, that are not competitive with respect to transferrin binding. So we obviously, did not want to disrupt iron uptake mediated through transferrin. And so what you can see in the green, blue, and gold color is the structure of CD71. What we've done is to look for binders to the blue region, which is termed the apical binding domain. So that apical binding domain does not make any contacts with transferrin. And so we've identified a number of Centyrins that bind to that domain and do not impact transferrin binding. We've actually also identified Centyrins that do block and bind to, on the right-hand side, the gold region. And those have been shown to actually block transferrin. So there may be other utilities for Centyrins that bind outside the non-apical binding domain. However, for the purposes of our studies, we're using Centyrins that bind to the apical binding domain. So despite their small size, as I mentioned, Centyrins are only 10 kilodaltons in size. So, they're 1/15 the size of an antibody. They're highly specific. And so what we've done here in collaboration with a company called Undruggable Molecular is they've actually expressed about 5,000 different cell surface receptors and soluble extracellular proteins on cells. And by flow cytometry, they can actually identify which proteins of those 5,000 proteins whether it's an antibody or Centyrin, can bind to. And quite remarkably, what you can see is that both the Centyrin alone on the left-hand side or the Centyrin siRNA conjugate only bind to CD71, as indicated by the blue dot in the upper right corner. So it's really a remarkable level of specificity. We've looked at antibodies, and many antibodies are highly specific, but as I'm sure you know, many antibodies can bind to multiple targets. So this really confirms that the lead Centyrin targeting CD71 is highly specific for its target. So the disease we're working on is a disease called Pompe disease. Pompe disease is one of the very rare glycogen storage diseases. What happens is that patients are deficient in the enzyme called GAA, which is the enzyme responsible for breaking down glycogen. And so what happens is you get an accumulation of glycogen within the lysosomes of the muscles of these patients. It's a very awful disease. And so if you get IOPD, meaning the infantile form of Pompe disease, typical life expectancy is less than 1 year if left untreated. LLPD also affects life expectancy, but more importantly, the quality of life. There's really only one approved therapy, and that's enzyme replacement therapy, which is not all that effective. So essentially, patients are treated with the GAA enzyme, but less than 1% of the enzyme actually makes it into the muscle and specifically into the lysosome to degrade glycogen. So our approach is to target an enzyme called GYS1, shown in the middle, and that's the enzyme that is absolutely responsible for glycogen production within muscle, both skeletal muscle and in the heart. And we believe by decreasing the level of GYS1 with our siRNA conjugate targeting GYS1 mRNA, we'll be able to ultimately decrease glycogen levels and restore them to a more normal balance within the lysosomes of patients with Pompe disease. And this is just one example of a data set. These siRNA Centyrin conjugates are highly effective, and more importantly, they have very long pharmacodynamics. And so the data I'm showing you here is in a Pompe mouse model. So it's a model where the GA enzyme has been knocked out. We gave a single dose, a 3 mpk of the siRNA Centyrin conjugate. And then 4 weeks later, we sacrificed the animals and looked for just one message and protein in the gastrocnemius, the skeletal muscle, and in the heart. And what you can see in blue versus the black, which is untreated, is that we get very profound knockdown of both the mRNA and the protein in both the gastrocnemius and the heart. So again, a single dose, very effective and very long pharmacodynamic half-life. And we've done longer studies and actually have activity out to 8 to 12 weeks. So we're moving our lead molecule forward. We hope to be in the clinic sometime later next year. And so obviously this is a very new scaffold. And so we wanted to do work to better understand the potential for immunogenicity. And so we work with our Proimmune colleagues, and I'm not going to go into the background because you just heard some great talks on the various assays that Proimmune has developed. And so we use the DC T cell assay in order to characterize whether or not there are any T-dependent antigens within the Centyrin scaffold. And again, it's 90% human matched to fibronectin or tenacin C, but there are residues that are mutated again to improve binding and alter specificity. What we found in the T cell activation assay that was described earlier by Andrew is that the 2 positive controls give a response index. It's very high, 60 and 32, but our lead CD71 Centyrin had a response index of just 0.19. And so the majority of approved biologic drugs have very low response indexes, less than 1. Again, I won't describe the assay since that has been well described. And so the low response index suggests that the Centyrin should have a low potential for immunogenicity. We've seen no evidence of induced rapid clearance of the drug in vivo, and we're just now in the process of developing highly sensitive ADA assays to characterize the potential or whether or not there are ADAs in our cyno and mouse studies. Importantly, historically, for the first 10, 15 years of development of oligonucleotides, one of the challenges was that the oligonucleotides induced innate immune responses that was overcome by various modifications to the nucleotides. So we've imparted those modifications on our siRNA. And so we also looked at cytokine release from human PBMCs and found that there was no cytokine release of our Centyrin siRNA conjugate. So we think it's quite inert with respect to either T-dependent immune responses or the induction of innate responses. So why is that? Again, it's not a fully human protein. There are these residues that are mutated. Again, I probably don't need to show this slide to this audience, but just to refresh your memory that class 2 antigens are taking up into an early endosome, which has a pH between 6 and 6.5. You get fusion of some of those early endosomes with the MHC containing endosomes. The pH drops a bit more, about 4.5 to 5, and then you get expression of the peptides that have been generated through processing in those early endosomes. And those peptides are expressed in the context of the MHC molecules on the cell surface. And so, again, our prediction was that, or our hypothesis was that Centyrins are very, very stable proteins, and we believe that that stability leads to an inability to actually process the centyrin within the early endosomes. And I'll show you data that somewhat support that hypothesis. So it was a really nice paper that was published, I guess now about 12 years ago. And in this study, they actually looked at the relationship between the melting temperatures of two proteins and their ability to induce an immune response as measured by antibodies. So they looked at hen egg lysozyme and also mouse lysozyme, knowing what the structure was, the two structures shown on the left. They were able to impart different mutations that did not completely unfold the molecules, but rather just altered their melting temperatures. And you can see the 8 molecules that were made in their melting temperatures. They were able to make mutants of the hen egg lysozyme that varied in melting temperature from the wild type by 25 degrees, and for the mouse lysozyme by 5 degrees. They then assessed those molecules in a mouse and whether or not there was an induction of anti-lysosome antibodies. And panel A is the hen egg lysozyme study. Panel B is the work on the mouse lysozyme. The y-axis is essentially a measure of the antibodies against either one of those antigens. And what you can see is that there's, in red, are the melting temperatures of the various mutants. There's a very nice correlation between melting temperature, in that the higher the melting temperature, the more stable the protein, the lower the induction of an immune response. Now, the absolute melting temperatures and absolute amount of antibodies varies between the two different proteins, but there's clearly a very nice trend in that, the more stable the molecule, the less of an immune response induced. So what's the data with our molecule? As I mentioned, centyrins are highly stable molecules. So in the left panel is the CD71 Centyrin alone. In the right panel is the Centyrin siRNA conjugate. Along the x-axis, what we looked at was the melting temperatures at various pHs, which would mimic the pH in either serum or in these early and late endosomes. So we've gone from pHs of 3.8 all the way up to 6.6 or 7.4 on the right-hand side. And what you can see is that the melting temperature is really quite stable across this wide range of pHs. Importantly, the melting temperatures are in the range of about 75 to 80 degrees, so slight variation, but extremely stable in high melting temperatures. So we believe that, getting back to this diagram, that when the CD71 Centyrin, or any Centyrin for that matter, is delivered into the early endosome, that there's really no disruption of the structure. And so there's no unfolding. There's an inability for the cathepsins or other enzymes to actually degrade the centyrin, and that it actually makes it to the lysosomes where, cannot get processed and presented in the context of MHC molecules due to this high stability as it traffics through the early and the late endosome. So I want to leave you with one last piece of data. So many of our centyrins, we initially purify them using a histidine tag. And we've always been concerned with whether or not a histidine tag could induce immune responses. Histidine actually makes it much more facile to purify these proteins. What I will say is the molecule we are taking forward does not have a histidine tag, but if you go back to the literature, there's actually very sparse literature, despite the fact that histidine has been used very widely to purify proteins, but sparse literature on whether or not histidine can actually be immunogenic. And so we did a study with Proimmune. Again, this is the DC T-cell assay, where we looked at two different centyrins, the CD7149, or the CD71-96, plus and minus a histidine. And what we found is, as you go across to the right, the percent antigenicity was really quite low, and really no significant differences between the plus or minus histidine. The strength of the response was similar, and then the response indexes were all below 1 and very similar to one another, regardless of whether the molecule had a histidine tag or not. So I think this is an area that requires, a bit more study, but given the fact that histidine can really facilitate purification of drugs, there may be value in further understanding whether or not histidine in the context of other protein scaffolds can actually be immunogenic. And if it's not, it may be incorporated into molecules in the future that do go into the clinic. So with that, just wanted to summarize that, again, we're using the Centyrin platform to deliver siRNAs into cells in a tissue-specific manner and outside of the hepatocyte. We have, very nice data showing we can deliver to muscle. And more importantly, we believe that we've de-risked the potential for immunogenicity of this very novel platform using assays that were developed by Proimmune and also the work that we're doing in vivo to assess ADAs and either neutralization or potential for rapid drug clearance in vivo. So with that, thank everyone. Thank all my colleagues at Aro. I'm happy to take any questions.
Fantastic. Thank you very much indeed, Steve. We do already have a first question from Melissa. And Melissa asks, what is the advantage of the Centyrin over a DARPin?
So, they're actually similar. I would say, Centyrins, based on what we see in the literature, might be a bit more stable. Again, we've identified 23 different sites to which we can engineer in a cysteine. So we have a lot of flexibility in this platform. But, I can't say anything negative about DARPins. They're a great platform as well, but we believe our platform might have broader utility in terms of delivering various payloads into cells.
Fantastic. Does anybody else have any other questions at all? Mike does. Excellent. Mike, do you want to unmute?
I just did. Hey, that's very cool. A couple of questions. If your Centyrin gets all the way to the lysosome, so therefore doesn't get out and be immunogenic, how does the SI survive that to be able to work?
Yeah, so it's a great question. So I didn't have time to show it, but we've actually done confocal imaging where we looked at trafficking of the siRNA Centyrin conjugate through the cell and actually compared it to an antibody conjugate. The siRNA Centyrin conjugate resides in the endosome for a long period of time, much longer than an antibody, so in the order of 6 to 8 hours. And so what happens within the early endosome is you get escape of the, well, we believe that you can either get escape of the antisense from the endosome, which can then bind to the RISC complex in the cytoplasm, or the entire conjugate can escape the endosome slowly. So, not all of the molecule goes to the lysosomes. Much of it stays and resides within the endosome for a long period of time to enable release of the siRNA.
Cool. One more question, if you don't mind.
Yep.
When you're not getting your 85% knockdown in the muscle, do you know if that's 100% knockdown in 85% of the cells, or if it's a partial knockdown when you're getting into all the cells?
Yeah, and it's a great question. It's a bit tricky to do, but we are going to try to do some in situ hybridization, so IHC, to see whether or not, doing FISH, whether, to your point, is it 85% across every cell or not? And, that's been done with some siRNAs in the liver, and the data are quite varied, but it's a really interesting question. It's something we want to look at in the future.
Cool. Thank you.
Yeah.
Thank you, Mike, as well. Any further questions? Well, perfect. Thank you very much indeed, Steve. Really appreciate that, and your insights into the technology. It sounds really exciting. So we wish you best going forward with that.